Hello everyone, welcome to part 2 of General Bacteriology. Today we'll continue with the previous lecture and we'll start with our discussion about the prokaryotic and the eukaryotic cell wall and cell structures, okay? Before that, let me revise yesterday's learnings. Yesterday we learned about the cell structure, okay? In the cell structure we said that there is cell wall, there is cell membrane, there is one nucleoid and also ribosomes. All right, now we'll just discuss the differences once. Prokaryotic cells or bacterial cells basically have cell wall made up of peptidoglycan, and eukaryotes, which basically includes fungal cell wall or human cell wall, it has does it have cell wall? In case of humans, no. In case of fungus, yes, it is made up of chitin. Okay, then we also discussed about the cell membrane, but today I'd like to ask, does bacteria have cell wall? Yes. Does eukaryote cells have cell wall? Yes. But the basic difference is bacterial cell wall does not have sterols, but fungal cell walls, they have sterols. Okay, they have sterols. So eukaryotic cell wall have sterols. Eukaryotic includes humans and fungus. Humans, they basically have cholesterol. Okay, cholesterol, and in case of fungi or fungus, it is ergosterol. You get the difference? Ergosterol. And as I said, bacteria do not have sterols, but nothing is that simple. There is one exception, and the exception is mycoplasma. Talking about mycoplasma, it is the smallest bacteria. It does not have cell wall. So, as a compensation, you can say it has sterols. Okay, so till now what we've discussed is bacteria have cell wall made up of peptidoglycan. They have cell membrane without sterols, and the exception to that is mycoplasma, which is the smallest bacteria having no cell wall and has sterols. Okay, now because of the sterols, this mycoplasma also gets a fried egg appearance. Fried egg appearance. Clear? So far we discussed the cell wall, cell membrane, and about the bacteria. As I said, they have ribosomes, but they do not have other structures or other organelles. But in case of eukaryotes, they have ribosomes along with other structures. Ribosomes plus other structures, okay? And talking about the chromatin or nuclear material or nucleoid, bacteria have nucleoid. They do not have no nuclear membrane, so they do not have nuclear membrane, but eukaryotes have nuclear membrane. That is why we say nucleus is present. Okay, this is what we learned yesterday. I think now we're ready for this table. So let me start with the cytoplasmic structures. Cytoplasmic structures are absent in case of prokaryotes, present in case of eukaryotes. Done. Ribosomes, 70S is present in prokaryotes, 80S is present in eukaryotes. Done. Cell wall, made up of peptidoglycan, and in case of eukaryotes, it may or may not be present. Not present in humans. Right. Present in fungi as chitin. Okay. Done. Then organ of locomotion and all, yesterday we discussed. Talking about the nuclear structures. Okay, nuclear structure is circular in case of prokaryotes, and it also has plasmids. But eukaryotes, they have a cell membrane, which is not present in bacteria. Okay, and we also mentioned about the reproduction part. These eukaryotes, they undergo meiosis, but these prokaryotes, that is bacterias, do not undergo sexual reproduction. They undergo binary fission. Now we'll start our topic of discussion with binary fission. What is binary fission? Now binary fission is nothing but one getting distributed into two. So this is one cell. If it splits into two, and subsequently this cell will split into another two, and this will also split into another two. So from one cell we get two cells, from two cells we get four cells, from four cells we'll subsequently get 8 cells. So it's a logarithmic type of exponential growth, right? So this logarithmic growth, or you can say exponential growth, is known as binary fission. 1 to 2, 2 to 4, 4 to 8. Clear? This is binary fission. Now, growth. Right. We said binary fission is the pattern of growth. No sexual growth or no sexual reproduction. Okay. And there comes a topic like doubling time. What is doubling time now? Now, I said from one cell we get two cells, and from two cells we get four cells. Right? So, this time required for one cell to be multiplied to 2. This duration from 1 to 2. Duration from 1 to 2 is known as doubling time. Clear? Now, bacteria have different doubling times. Obvious, right? So let's do some comparison. E. coli multiplies in 18 minutes. That is, doubling time is 18 minutes. Roughly, you can say 18 to 20 minutes. Then MTB, that is Mycobacterium tuberculosis, it is 18 to 20 hours. And Mycobacterium leprae, it is 18 to 20 days. You get the difference, right? One bacteria, E. coli, can multiply or get doubled in minutes. Tuberculosis, Mycobacterium tuberculosis, it multiplies in hours. And leprae, on the other hand, multiplies in days. Okay, so you just think and tell me, if we do any culture for Mycobacterium leprae, that is, for example, a patient comes to you, okay, a patient comes to you with skin problems, nerve damage or something, then you do some skin smear, and then also you decide to do culture. Okay, then if you decide to do culture, you think you'll get result in one day for Mycobacterium leprae? No. Will you get culture in 2 days? No. So bare minimum you'll need, minimum there is 20 days for one doubling. Okay, that means for one bacteria to become 2, minimum 20 days is required. So it's long. To find out the culture report or culture is positive, it takes weeks. Okay, in case of leprae. Why? Because the doubling time is 18 to 20 days. But on the other hand, in case of E. coli, you get result within hours because it multiplies within minutes. Clear? So this is all about doubling time. Now let's just talk about growth. What is growth? For humans, this is growth, right? But for bacteria, being able to multiply is growth. Okay, so there are two scenarios. This is a state of surviving or living or being alive, and this is a state of growth, and this is the state of being dead. Okay, dead means, you know what dead means, right? Not surviving. And alive means just living. And growth means multiplying. I hope the concept is clear. Growth means being able to multiply. Okay, so how do bacteria grow? Okay, if you consider this to be the growth, okay, and this to be the time, okay, bacteria grow like this. Okay, now I'll describe each phase. This is the lag, log, stationary, and decline phase. Now, bacteria grow in 4 phases. Okay, number 1 is the lag phase. This type of phase wherein this is the bacteria. Okay, this bacteria now has to grow, so it becomes bigger in size. So in lag phase, bacteria becomes bigger. Lag phase, bacteria becomes bigger. How does it become bigger? It accumulates all the nutrients required for multiplication and it becomes bigger. Now you tell me, what will happen after lag phase? It has already accumulated all the nutrients. So the next phase is log phase, where it can successfully multiply. Right? It can successfully multiply. This is the log phase. So in log phase, you'll find rapid multiplication. Clear? Then next comes a phase where third phase where bacteria's, now bacteria started like this. They were small in size, they became bigger in size, right? They became bigger in size and then they started multiplying. Now after life comes death. So after multiplication there will also be a phase where bacteria start dying. They start dying, but remember, they are also growing, right? They are also growing, so growing or multiplying. So in this phase, the number of bacteria multiplying is equals to the number of bacteria dying. So the sum total is constant. You get my point? So some bacteria are multiplying, some bacteria are dying, but the net result or net is constant. Net constant. Okay, and fourth phase is all the bacteria die out. All the bacteria die out. Clear? So we discussed 4 phases. Number 1, where they become bigger in size. Number 2, where they multiply. Number 3, where they start to die. And number 4, proper death. So if we plot it like this is the bacterial, for example, if this is the bacteria number, okay, in first phase, this is time. Firstly, bacteria number is static, they just become bigger in size, lag phase. Then they start multiplying, so their number increases, right? Their number increases, so their number increases in log phase. Then there comes a phase wherein the number of bacteria alive and the number of bacteria dying, they are the same, so this is the stationary phase. Then comes the phase where bacteria start to die more, so decline phase. Okay, you get it? Decline phase. Now antibiotics. What are antibiotics? Antibiotics are drugs which kill bacteria. So they are more active. They can kill bacteria when the bacteria is multiplying. Okay. So in which phase do you think antibiotics will work? Antibiotics best work in the log phase. This is the phase for antibiotics. Also, if you want to see any biochemical test or staining, in this log phase, you get uniform staining also. Okay, then in stationary phase, this is the phase where the bacteria start to die. Okay, so in this phase, bacteria want to survive, so they form some structures which are resistant to bacteria or resistant to death. So resistant structures, also known as spores, are formed in stationary phase, and in decline phase they die. Okay, in stationary phase they turn into resistant structures, they form resistant structures called spores, or gram positive bacteria may also get converted to gram negative bacteria to survive better, for better survival. Okay, now we'll discuss about these again. Antibiotics, as I said, they work more in the bacteria which are multiplying. So they work in the log phase. They do not work in the lag phase. Antibiotics do not work in the lag phase, they work in the log phase. Then, in the stationary phase, as the bacteria start dying, they realize that they need to form resistant structures. So they form spores. Okay, also gram positive bacteria gets converted to gram negative bacteria. I'll tell you the theory behind it in some minutes. Then in decline phase, the bacteria actually start dying. Okay, um, and this is how their growth is seen. But do you think bacteria are all good? Okay, do you think bacteria are all good? No. The thing is, in between humans, is there any competition? Yes. So in between bacterias, will there be any competition? Yes, again. Okay, so bacteria, if this is one bacteria and if it wants to survive in stationary phase, what actually happens is that uh, the nutrients, like firstly in log phase, okay, firstly lag phase, in lag phase the bacteria accumulates lots of nutrients. Okay, it becomes bigger in size. In log phase it actually multiplies. Okay, because it has already accumulated stuffs, it can multiply now. And after it starts multiplying, what happens? Nutrients are consumed. Nutrients are consumed and metabolites accumulate. This is the reason why some bacteria start dying. Why? Because nutrients are getting over. Nutrients are getting consumed and metabolites start accumulating. So some bacteria start dying. And others are still growing, so it is stationary phase. Okay. And then when the nutrients and metabolites accumulation, toxic metabolites are in greater amount and nutrients are very least, then they actually die. All of them die, right? So in stationary phase, you think I just mentioned about this jealousy thing, in humans there is jealousy, in bacteria also there is jealousy. In stationary phase, once the bacteria realizes that there are lots of metabolites formed and nutrients are getting over, that nutrients are getting consumed rapidly, they say that let me survive and let him die. Okay, so the bacteria, what they do, they produce toxins. Okay, toxins which can harm other bacterias. So they produce toxins which can actually kill other bacterias. So in this stationary phase, one is that they form resistant structures so as to prevent death. Then they also can get converted to negative bacteria. Then third and important point, they form toxins, basically exotoxins, which can harm other bacterias. And antibiotics can also be obtained from bacterias, right? Antibiotics are basically synthesized. They can be obtained naturally, they can be obtained artificially, right? These naturally obtained antibiotics, these are nothing but substances which are secreted by one bacteria to kill another bacteria. You get my point? So there are some antibiotics which can be obtained from the stationary phase. These are nothing but toxins or exotoxins or antibacterial substances which one bacteria produces to kill another bacteria. Clear? So let me discuss again. Bacterias, they undergo binary fission. What is binary fission? They multiply into 2. 1 multiplies into 2, 2 multiplies into 4, 4 into 8, right? 8 into 16, exponential growth. Okay, like this. And their growth is basically in 4 phases. In lag phase, they become bigger in size. In log phase, they actually form lots of bacteria. In stationary phase, they start to die. Okay, and in decline phase there is actual death. Right, again, if this is the number, if this is time, initially there is lag. Okay, then bacteria start to grow in number, so number increases. Then, okay, they become, the number of bacteria which are multiplying and the number of bacteria which are dying become the same. So there is constant viable count. Now I'll change the term. Viable count is constant. Total count obviously increases. You get my point? Total count increases. That is, new bacteria are being formed. But since bacteria is also dying, right, bacteria also start to die, the viable count is constant. Then after more number of bacteria start to die, viable count also decreases. The viable count decreases. You get my point again? So I'll draw two graphs now. This is the total number of bacteria. This is the viable number or viable count of bacteria. And this is time. So initially, in both the scenarios, lag phase, then log phase, then total count increases. Okay, if you understand, total count increases, but in case of viable count is constant and viable count declines. You get my point? This is lag, log, stationary growth is like this, stationary, then decline. Okay, this is decline. Why this is straight line? Because no new bacteria are getting formed. Okay, in decline phase. So if you see the total number of bacteria, the growth is like this, but if you see the viable count, the growth is like this. So mind you, when I mention the count, it is always the viable count, not the total count. Total count will follow. So if I do both the viable and total count, it will be like, this is lag phase, then log phase, then total count will be like this, viable count will be like this, then this is decline phase, this is decline phase. I hope you understand this. You understand why the total count increases? Yes, because number increases, and after and during the decline phase, the number is not getting increased, so the number is constant. But in case of viable count, in the stationary phase itself, the count is constant. Why is it constant? Because the number of bacteria alive and dying are the same. And in decline phase, in case of viable count, it decreases because living count decreases. Okay, and this is total count. And this is the viable count. I hope I could make you understand. So growth is done, multiplication or like everything is done. Now I'd like to explain something. Cell structures, yesterday we discussed about it. Now I'll just mention the essential structures and non-essential. Okay, just a rough mention. Now what do you think is essential structure? Cell wall, definitely. Membrane, definitely. Then nucleoid, definitely. Then ribosome, definitely. And one else, mesosome. Okay, what is mesosome? Mesosome is invagination, which helps in transport. Okay, and then non-essential structures, if I may list, it will be capsule. Yesterday I discussed capsules. What are capsules? Capsules are basically structures like this, definite structures. And then there can also be glycocalyx. Okay, staph, epidermidis, very good example. And about capsules, I said all those organisms affecting the brain and lungs. So lungs, streptococcus pneumoniae, streptococcus pyogenes for throat, then Haemophilus influenzae, And for brain, I said Neisseria meningitidis. Okay, other structures can be pili, flagella. Okay, and what else? These are the non-essential structures. So you may later on also add biofilm and others. So capsule, glycocalyx, pili, and flagella are non-essential structures, and these are the essential structures. So today I hope we covered a lot. Did we cover essential and non-essential structures? Yes. Did we cover growth? Yes. The next topic is antibiotics. A very interesting and important topic. Okay. Now let me start. Antibiotics. Did I say that bacteria have cell wall? Yes. So we would like to kill it. Okay, through this cell wall mechanism. Now, cell wall, yesterday I said they are made up of peptidoglycans. Cell wall is made up of peptidoglycan. And how is this peptidoglycan made up of? Is it NAM-NAG subunits? NAM-NAG subunits. Did I say it or not? Yes. How are they connected now? They are connected by beta 1,4 glycosidic linkage. So, is there anything that would destroy this linkage? Yes. Enzymes like lysozyme. Lysozyme. Where is this lysozyme found? Very easy example, saliva. So, if you remember, when we were small, if we would get any wound or any attack, like any injury, any wound, what would we do? We would apply saliva. Would we not? Yes, we would. Why would we? Because saliva had or has lysozyme. Lysozyme can destroy beta 1,4 glycosidic linkage. So if beta 1,4 glycosidic linkage is destroyed by lysozyme, then the bacterial cell wall is gone. If the bacterial cell wall is gone, water will enter into the bacteria and the bacteria will swell up and burst. So there is bacterial death. So it is bactericidal. Okay, lysozyme is bactericidal, which breaks beta 1,4 glycosidic linkage. Okay, now let's talk about cell wall inhibitors. So I said cell wall is made up of NAM-NAG subunits, and yesterday I mentioned this NAM or muramic acid has alanine, glycine, lysine, alanine, etc. Then also it is connected to another AGLA subunit like alanine. L-lysine and alanine are connected by one linkage, which is tetrapeptide in nature. So there is one enzyme named as transpeptidase, which would modulate this action. Did I mention it or not? Yes. So there are drugs which target this transpeptidase enzyme, and these are penicillin, cephalosporin, or you may also call beta lactam drugs. So beta lactam drugs basically target this transpeptidase enzyme. Okay, beta lactam drugs target this transpeptidase enzyme. So talking about cell wall inhibitors, I mentioned beta lactam drugs. Beta lactam drugs are nothing but penicillin, cephalosporin, which target the transpeptidase enzyme. They also inhibit or bind to the penicillin binding sites. Okay, there are certain sites in the bacterial cell wall like penicillin binding sites. Okay, when these beta lactam drugs bind to penicillin binding site, they alter the symmetry, they alter the membrane structure and cause influx of water or any like fluid and cause lysis of the bacteria. Okay, so beta lactam drugs and also normally during cell that is multiplication, bacteria release autolysins. Autolysins are enzymes which basically regulate this growth. Okay, but when the beta lactam drugs are given, autolysins are always active or activated, which causes self destruction of the bacteria. So three mechanism by which beta lactam drugs or cell wall inhibitors act. So talking about cell wall inhibitors again, I mentioned beta lactam drugs which act by three mechanisms. Number one is transpeptidase. Number two is like, firstly they inhibit transpeptidase. Number two, they bind with penicillin binding sites. Number three, they activate autolysins. These three mechanisms for all beta lactam drugs. Okay. Then, are there other beta lactam drugs? Yes, definitely. Vancomycin and others. Okay. Now, how does this vancomycin act? So again, going back to that AGLA structure. Firstly, when nascent cell membrane is being formed, okay, this agla, which is like actually 4 peptide, now initially it is 5 in nature. There is one terminal alanine, okay, which separates out, and when it separates out, okay, when it separates out, a bond can be formed with another AGLA structure, L structure. Okay, so basically when it is nascent, there is one extra alanine attached to the 4th peptide. Okay, when the cell wall is nascent, there is one alanine attached to the last alanine. That is, instead of 4 peptides, there are 5 peptides. Okay, but when it is like after being, firstly though it's nascent, but later on this bond is broken and another bond is formed with lysine through transpeptide enzyme, right? But this vancomycin is one such drug which does not allow the breakage of alanine-alanine bond. Okay, so these D-alanine and D-alanine bond is not broken because of vancomycin. Vancomycin prevents the breakage of terminal D-ala bond. Vancomycin prevents the breakage of terminal D-ala bond in nascent peptidoglycan side chain. So what is the result? Because this bond is not broken, now this alanine cannot form bond with another lysine. As a result, there is no cross link formed. If there is no cross link formed, yesterday also I mentioned, if there is no cross link formed, there will be no stability. If there is no stability, bacterial cell wall will definitely rupture. Clear? So bacterial cell wall is made up of peptidoglycan. Peptidoglycan has NAM, NAG, NAM, NAG subunits. NAM has agla. Okay, it's terminal, like NAM as its side chain. Now this agla, the last A is always attached with another A in the beginning. Okay, which is the nascent form. But later on, this bond is broken down and alanine forms bond with lysine of another side chain, and cross linking is done, stability is provided. But if this alanine do not separate, what happens? That the cross linking is not able to be formed, and because of that because of lack of cross link, there is no stability. Got it? So vancomycin works by that mechanism. Cell wall inhibitors, I mentioned beta lactam drugs. Okay, beta lactam drugs are nothing but like having a thing like this, having a ring, beta lactam ring looks like this. Okay, so drugs having one beta lactam ring are known as beta lactam drugs, which includes penicillin, cephalosporin, etc. Okay, they act by three mechanisms. One, they inhibit cross-linking. Number two, they bind with the penicillin binding site, binding protein. Third, they activate autolysins. autolysins. Then we looked into other cell wall inhibitors like vancomycin. How do they act? They prevent the breakage of D-allocyte chain. We looked into two antibiotics cell wall inhibitors. Now, cell wall inhibitors are done. Then we'll look into cell membrane inhibitors. Okay, cell membrane inhibitors. Cell membrane inhibitors basically include polymyxin and daptomycin. Just remember the names, polymyxin and daptomycin, PD for now. Then there will be some protein synthesis inhibitors. So protein synthesis inhibitors. What are protein synthesis inhibitor? Ribosome, as you know, has 30S and 50S subunit. So there will be some antibiotics which act on the 30S subunit and prevent protein synthesis. There will also be some which act on the 50S subunit and prevent protein synthesis. The ones acting in 30S subunits are aminoglycosides and tetracycline. How will we remember? At 30. Okay, so aminoglycosides and tetracyclines, they act at 30S subunits. Okay, and the rest are 50S. Rest includes clindamycin, linezolid, macrolides, clarithromycin. Clear? So protein synthesis inhibitors are done. They basically prevent the synthesis of protein by either acting on 30S subunit or 50S. 30S we remember by the mnemonic at 30. A for aminoglycoside and T for tetracycline. Okay. Then lastly, nucleic acid inhibitors. Nucleic acid synthesis inhibitors. So there are some which inhibit DNA, some which inhibit RNA, in some which inhibit the nucleotide. Okay, so nucleotide includes quinolones, clear. Okay, and DNA synthesis inhibitors basically DNA synthesis inhibitors basically include quinolones. Okay, quinolones. RNA synthesis inhibitors basically include rifampicin. In nucleotide synthesis inhibitors basically include sulfonamide. Okay, trimethoprim, triamterene. Okay, sulfonamide for example. So sulfonamide for nucleotide, rifampicin R for RNA inhibitors, and quinolones for DNA inhibitors. I'll tell you the mechanism in detail in subsequent lectures. Why not now? Because it will be overloaded. You'll be overloaded, but later on I'll prepare a lecture where I'll discuss about nucleic acid synthesis inhibitors, mechanism in details. Whenever we come to that bacteria and the drugs acting on it, we'll discuss the nucleic acid synthesis inhibitors also. For now, just remember quinolones for DNA, rifampicin for RNA, sulfonamides for nucleotides. And about proteins I said already. Then about bacterial, cell wall and membrane I said already. So which of these antibiotics are bactericidal, that is can kill, and bacteriostatic, which can inhibit growth? Okay, cidal, the cell wall and the ones acting in the cell membrane, they are always cidal. Why? Because obviously the membrane is getting damaged, the wall is getting damaged. So what happens is that water or fluid will get inside the bacteria and the bacteria will rupture. So they are always cidal, common sense. And about protein synthesis, only aminoglycosides are cidal, rest all are static. And then nucleic acid synthesis inhibitors. Okay, these quinolones, DNA, RNA, the ones which affect DNA and RNA, they are cidal. And the one affecting nucleotide, it is just one base pair, right? So they are static. So quinolones and rifampicin are cidal and nucleotides are static. Clear? So I gave you one easy way to remember also. We'll talk about the mechanism of nucleic acid synthesis inhibitors in details in subsequent lectures. For now, just remember about cell wall synthesis inhibitors because they are very important. There are lots of resistance in beta lactam drugs. Okay, so today we studied a lot. I hope I'll be able to cover other topics in subsequent lectures. Thank you so much.